Hey everybody, we're going to do our notes on muscle responses. There's only 11 slides, so hopefully this one won't take too long. But I put a question up on Google Classroom about potentially hosting a Zoom call or a Google Meet to see if people would be interested in doing that before we take a test or a quiz the next time. We actually did a Zoom call with Mr. Robertson today, and it worked really well, and it was kind of good to see some of our teachers and talk to him in person instead of it being just a video. So if y'all will respond to that question, that would be awesome, and I'll figure out a time to Google Meet or Zoom call with everybody, and it'll just be if you're interested. You're not required. I just want to see y'all if you need to have some questions answered or for us to have a little bit more of a conversation. All right, so today's notes are on muscle responses, and this is going to be all about creating a graph of how a muscle contracts. And so the first thing we're going to talk about is the stimulus that is required for that muscle contraction to occur. And the stimulus can be sub-threshold or under threshold, and the muscle won't contract. So it would be like you trying to start your car and it's just not happening. And no matter how many times you turn the key, if that battery's dead, then the car won't turn, or the car won't turn on. And that's how it would be if the stimulus is underneath the threshold stimulus or below the threshold stimulus. The threshold stimulus is the smallest stimulus that will cause the contraction of a muscle fiber. Now notice that says muscle fiber, that does not say muscle. Okay, so even if we compare this to the bicep, that whole bicep may not contract at one time. In fact, it very rarely does unless you're pushing it to its maximum capacity. This is where, or the threshold stimulus is the stimulus required for a muscle fiber, a muscle cell to contract. And if you know the steps of muscle contraction, this would be when that acetylcholine is rushing through the synaptic cleft, binding to the sodium ligand gated channels. If there is enough sodium that rushes in to that muscle fiber, to that muscle cell, to turn an action potential into a muscle impulse, then you have reached threshold stimulus. If you have not, if you don't have enough sodium rushing in, then you would be down here on the graph. You would be below the threshold stimulus. But here, this peak on the graph represents the potential across the sarcolemma. And when you get past this red dotted line, you do contract, and that muscle fiber will contract. And this is an all or nothing response. Once you tip the domino down and that domino effect spreads down the sarcolemma, that muscle fiber will contract. There is no break in the middle. There is no half contraction. If you turn an action potential into a muscle impulse, then it will contract. And the all or nothing definition is the idea that if the stimulus is above threshold, the muscle fiber will contract completely. If it's under it, then it won't. Recruitment. Okay, so recruitment is all about recruiting more muscle fibers to produce more force and produce more contraction out of the entire muscle. I was reading in the textbook today, and then there was a picture that kind of caught my eye, and it was a guy that was eating an ice cream cone. And I thought, well, that's a really good analogy for, a really good example for how a small amount of muscle fibers that are controlled, or a small amount of muscle fibers when they are stimulated could bring your hand to your mouth to eat ice cream. But if I'm trying to lift a weight, or I'm trying to lift something heavier, then I have to recruit more muscle fibers to contract to get more work out of that muscle. And so you don't contract the whole muscle every single time you bend your arm. If you think about your bicep, if I just need to eat ice cream, I don't need a lot of work out of the bicep. But if I'm lifting something heavy, then you are going to recruit more motor units, therefore more muscle fibers. If you look at this picture and compare the colors, it'll help you out. So in this picture, we're just looking at muscle cells here, and we'll start with motor unit 1. Okay, a motor unit is a motor neuron and all of the muscle fibers that it controls. So if you only stimulate motor unit 1, you don't get a lot of force out of the muscle. Okay, that could be your ice cream cone. If you stimulate just motor unit 2, then that would be a little bit more force. Maybe that's like lifting your textbook. But if you recruit and you send a nerve impulse down motor unit 1 and motor unit 2, then you get a lot of force out of that muscle in comparison to just 1 or just 2. So this would be lifting something even heavier. And that's going to be correlated with low-level stimulation and high-level stimulation. Just think about what that says. Low-level stimulation, I don't need a lot of motor units. I'm just giving the muscle a little bit of stimulation to contract. High-level stimulation would be additional motor units are activated. You're recruiting more to do more work. A twitch contraction is a just single contraction and relaxation of a muscle. Now I am talking about the whole muscle in your body, the whole bicep, the whole, what did we do yesterday? Rectus femoris in your thigh. Okay, so these are all really produced in the lab. It would be hard to do this outside of the lab, but the reason that they do this is to see what kind of relaxation and contraction periods does that specific muscle show on a graph. And I have one on YouTube here to show you, and this is kind of a gross lab, but most colleges will do it in physiology, but you take a frog leg and you remove one of the quadriceps or hamstring muscles out of the frog leg and you zap it with a little bit of electricity, and you can see the quadricep muscle here fully contract and relax. So let me start it over for you and you can see. Okay, and it doesn't work long because you have to have the leg in a saline solution, and once that muscle's out of the body for a short period of time, it loses its ability to contract. But that's what it would look like. If you give a muscle enough stimulation with electricity, it's the same thing as that motor neuron telling it to contract, and you can measure that in lab using something called a myogram. Okay, and a myogram is the graph of the force of the contraction over muscle of a muscle over time. So when you look at this graph, we've got tension or force on the y-axis, and you have time on the x-axis. And this is only 0.1 seconds. And in this graphical reading of a muscle, you can see where the stimulus was applied. That's your little arrow right here. There's different periods that we're going to talk about of a myogram, which indicate what happens before contraction, what happens during contraction, what happens during relaxation, and then the process can start over. Okay, so make sure you label these on your graph, and now what I'm going to do is talk about what each one of them is. The first part of a myogram has to be the application of the stimulus. If you don't have a stimulus, you don't have contraction, or you at least don't have the green light to contract. The second part is called the latent period. This is right before contraction. Then you'll have the period of contraction and the period of relaxation. Now what we'll do now is go through the steps that are going to happen during the latent period, the steps that are going to happen during the period of contraction, and the period of relaxation, and how that relates to the notes that we did a couple days ago. Okay, the latent period comes first. Okay, this is after the muscle has been stimulated to contract, and the stimulus is there. So the sodium has rushed in, but the latent period is the short period of time between the sodium rushing in at the motor end plate and then that impulse spreading down to reach the T tubule. So again, there's just a small lag of time where we've knocked the dominoes down and we've got to wait for the dominoes to fall down into the T-tubule. So again, the motor end plate is just where the receptors are found, the sodium ligand gated receptors. That sodium rushes in, that is an action potential. We have the potential to do action. We have the potential to turn into a muscle impulse. And this latent period is just that lag before that impulse travels down the sarcolemma, specifically to reach the openings of those T-tubules and tell that terminal cisternae to release calcium into the sarcoplasm. Now, once that calcium is into the sarcoplasm, it's come out of the voltage-gated calcium channels of the terminal cisternae. It's floating in the sarcoplasm. Now, in the period of contraction, we've got to contract. And the steps that cause contraction are calcium binds to troponin, that shifts tropomyosin out of the way. The sites on actin are exposed so that actin and myosin can click together. They can hook together. The head of myosin is going to link up to the little bitty bead on actin so that you can have the power stroke, the myosin head can flex, and that sarcomere can shorten. Once the sarcomere has shortened, that is the end of contraction. Now we got to figure out, well, how do you undo everything to get me relaxed, get the whole muscle relaxed so that I could contract again if I wanted to. That period of relaxation requires ATP to put calcium back into the cisternae. Remember that it's going against its concentration gradient. When calcium unbinds from troponin, tropomyosin shifts back. Actin and myosin can no longer form a cross bridge, and everything slides back to its resting point. Notice that that period of relaxation is longer than the period of contraction. Okay, so it takes a little bit longer to relax the muscle, and that is normally due to the fact that it takes a while to get that calcium back into the terminal cisternae. For example, in your textbook, it says that the period of contraction is 10 to 100 milliseconds. So therefore, the period of relaxation would be a little bit longer. Let me see if it says anything else. Okay, no. All right, let me keep going. Now, that's the end of the myogram. And really, this is just a labeling representation of the steps that you already know about the muscle contraction and relaxation, and just understand what those mean. You know, that the latent period is just the spreading down the T-tubules to release calcium. From that point on, we have the period of contraction when we are contracting the muscle, shortening that sarcomere. And then the period of relaxation would deal with ATP putting calcium back into the terminal cisternae, and even the acetylcholinesterase breaking down acetylcholine so that we can stop that action potential altogether. In Muscle Man, I'm going to talk about these three terms, and I just want to go over it since we're in the muscle unit. But muscles like to work together. It's a lot easier on them if they have buddies that work with them instead of having to do all of the work, which is why we have 300 and something pairs of skeletal muscles. But they are split up into three general types, and the first one is the prime mover. And in Muscle Man yesterday, we did rectus femoris, and the rectus femoris is the prime mover for your quadriceps. It's the one that does most of the work, but it also has synergists that work with it. They're going to assist or help out the prime mover. And in your quadricep group, there are 4 muscles, hence why it's called quads. And the rectus femoris, again, is the prime mover, indicated with this red arrow, but the synergists are going to be on either side of that and are going to help aid that prime mover so it doesn't wear out or fatigue easily. The antagonist is going to be the muscle that is going to resist the movement of the prime mover. So if we look at the leg, the quadriceps are on the front of the thigh. The hamstrings are on the back of the thigh. And if we were just talking about the entire group, you could say that the hamstrings are antagonistic or antagonist to the quadriceps. And remember, muscles move joints. They stabilize joints. They don't just, our movement doesn't just happen unless a muscle is pulling on a bone like a lever. And so you need one on either side of that joint to pull it back and forth. If you contract both of them, okay, which would be hard, but if you contract both the prime mover and the antagonist, nothing happens. And I was also reading in your textbook today about how muscles are designed to contract at low level stimulations to produce posture or to help you stand up. If that required a high level stimulation, or if that required all of the motor units to be activated, then that muscle would wear out really quickly and would fatigue and would require more ATP than it should. And so the purpose of having those motor units is really just to save the muscle some energy, so that you don't have to have as much ATP produced to just stand up or to just sit or to just rest your feet on the couch, for example. All right, so that's the end of the notes for today. Let's see where the green sheet is. I think y'all might have a review sheet on this. Let me see here. Oh no, tonight for homework, you only have to do the rigor mortis reading, which is the stiffness of death that occurs because of a lack of ATP. So just read that, turn in those questions like normal, and if you're running into any issues with Google Classroom, just let me know.